So, as I said, the amino acids, once they've been brought together, a peptide bond will form between these, and now they are joined. So, step 5, the ribosome then moves along one codon. So this whole ribosome is going to move along one codon, and probably in this direction, and the tRNA releases amino acids and can be reused to collect another amino acid. So the tRNA, which is this thing here, was attached to this blue amino acid, but now that this amino acid is bound to this one by a peptide bond, this tRNA is now free. It leaves the ribosome and it will go and collect another amino acid and bring it back. So these tRNA basically they can be reused, which is very useful. And part 7, steps 3 to 6 are then repeated until it reaches a stop codon. So basically, this keeps happening. The ribosome is moving along the mRNA, moving along from codon to codon to codon, and each time it does that, the tRNA with an anticodon that base pairs with the codon will come, base pair, and they will bring whatever amino acid they code for. Those amino acids are then one after the other joined together by peptide bonds until you basically have something that looks like this, and that would go on much longer, and you've got a long chain of amino acids bound together by peptide bonds. This is called a polypeptide because poly means many, so many peptides, okay, and this eventually will make up the protein. The process stops, as I said, once you get to this, which is called a stop codon, and that just stops the process. Now, the last little bit of this key area, which I should get in this video, just go through this. As it says here, one gene, many proteins. The same gene can be used to make several different proteins, and there are two main processes as to why: alternative RNA splicing and this thing called post-translational modification. Now, this second one here is actually being removed from the higher course in the last couple of years. I've kept it in here just so you're aware of it if you ever see it in past papers, but we don't actually need to go through that because, as I said, it has been removed. So we're only going to focus on this first one, alternative RNA splicing. Here we go. So under certain conditions, alternative segments of RNA may be treated as exons and introns. So sometimes, depending on the conditions, an intron might be an exon in certain circumstances. So different regions of the gene can be coding at different times. So we have a look at that as a diagram. Here is our primary transcript, alternatively spliced mature mRNA transcript. So sometimes these might be the exons which are kept. Sometimes these could be exons which are kept. Obviously, once they're translated, this would result in different proteins being produced. So in other words, one gene can produce several different mature mRNA transcripts and therefore several different proteins.